So I'd like to introduce our first speaker for this session. This is Professor Peter Donnelly. Professor Donnelly is also the director of the Wellcome Trust Center for Human Genetics, as well as Statistical Science Fellow of St. Anne's College. He is well known for his work in molecular evolution, tracing the roots of human existence to their earliest origins using the mutation rates of mitochondrial DNA. And he studied genetic distributions in living populations to trace human evolutionary history, which informs research in evolutionary biology as well as medical treatment for genetic disorders. He's a key player in the international HAP-MAP project and the ongoing international effort to model human genetic variation and pinpoint the gene responsible for specific aspects of health and disease. Also, he's a leading expert on DNA analysis and the use of the forensic science in criminal trials, and is an advocate for bringing sensible statistical analysis into the courtrooms. And his topic today is the genetics of personalized medicine, challenge and opportunity. Professor Peter Donnelly, please.
Thank you very much. It's a great pleasure to be here in Macau, and it's
好多謝今次基金會請我嚟澳門啦，咁係好精彩嘅演講。咁尤其是係禤先生同埋何鴻燊基金會醫療拓展基金會啦，咁係一路喺個人化醫學嗰度係做咗好多嘅支持啦。咁係一路喺幫咗好多喺大眾啦，或者係喺病人啦、醫生啦，或者係醫療嘅團隊嗰度咧，係做咗好多嘅醫療教育同埋推廣。咁係喺呢度喺好多謝佢啦。除此之外呢，亦都係令到我哋幾個團隊係多咗好多嘅交流啦。譬如琴日我哋喺香港做咗一啲嘅交流啦。亦都包括中文大學嘅團隊啦。譬如盧英明教授啦，亦都係做咗我哋嘅advisor 啦。咁係禤先生同埋基金會係俾咗我哋好多支持之外咧，係促進咗我哋好多嘅多方面嘅交流。I'm very grateful for Patrick's friendship and for Isabel's friendship and their family, which has been a wonderful thing over the last few years. 咁而最後就係話，除咗交流之外，亦都建立咗好多嘅友誼啦。咁而呢樣嘢我係我哋建立咗嘅友誼係令到我好開心嘅。咁頭先Donnelly教授講過話一啲喺個人化醫學喺腫瘤方面嘅一啲嘅發展啦。腫瘤當然係一個基因嘅有關嘅病啦。咁我希望可以講一講基因呢係點樣可以幫到我哋其他病嘅診斷啦。 或者係預測啦。咁點樣或者係我哋嘅基因點樣可以幫到我哋去發展一啲新嘅藥物啦？咁而我自己本身係一個統計嘅學者咧，咁希望點樣可以用一啲數學或者係基因嘅模型咧，係將基因嘅資料帶到去一啲嘅應用方面。咁而大家可以見到啦，咁我哋過去嗰幾十年，我哋係喺基因方面嘅資料係多咗好多啦。咁可以見到2000年嗰陣時係一啲基因嘅排序係技術好貴啦。咁所以唔係話好多嗰時，仲唔係話好多嘅基因資料係我哋可以揾到啦。咁但係就一路咧係喺人類基因嘅定序測序嗰陣時係有好多嘅進步咯。咁所以我哋可以見到喺我哋擁有嘅一啲基因嘅資料嘅數目方面呢，係有一個爆炸性嘅提高嘅。咁而家喺呢一點，可能成個地球上可能有10萬至到20萬嘅人係基因係測序咗啦，我哋知道晒佢基因嘅排序啦。咁而我哋都期望喺未來嘅時間，可能係好快就會超過100萬。咁而好可能喺10年或者15年之後，會有10億嘅人係我哋係知道晒佢基因嘅排序嘅。咁而呢啲資料係會喺兩個層面嗰度會應用到。第一，可能佢會係喺一啲大嘅數據嗰度咧，係同一啲臨床數據整合咗啦。咁第二就話可能會喺大家嘅生活上，甚至乎係大家電話啊、大家手錶都可能係會揾到關於你自己嘅一啲嘅基因排序嘅數據啦。咁而因為個數據係咁大啦，咁所需要嘅電腦啦、數學嘅模型啦，或者係一啲電腦儲存啦，或者分析嘅能力咧，係好重要嘅。咁而我哋頭先亦都提過啦，一啲藥物嘅發展啦。咁我哋可以見到其實發展一隻藥其實好困難嘅。咁而喺過去幾年，我哋見到咧，我哋成功發展一隻藥物嘅數量咧係越嚟越少嘅。咁而因為做一隻新藥其實越嚟越貴啦，需要好長嘅時間啦。咁可能係要做好多嘅病人嘅數據先可以真係去到市面上係出現啦。咁所以做一隻藥係比以前困難咗啦，需要更長嘅時間啦，需要更多嘅金錢資源啦。咁而可能亦都要有越嚟越多嘅臨床數據真係試用過，我哋先可以將隻藥帶到去市面嗰度。咁而我哋亦都知道啦，有咁而好多藥其實喺中間呢係失敗咗嘅。咁而可能講緊話第10隻藥入面，真係做一啲臨床前嘅發展。而到最尾去到一個臨床研究呢，只不過10隻藥入面可能得1隻啦。咁而臨床嗰度亦都好多藥呢係去失敗咗嘅。咁而因為咁樣呢，嗰個成功率咁低呢，我哋知道每一隻藥平均去發展成功係會花咗10億至到20億美金，先至係可以成功地將一隻藥帶到去市面嘅。咁 其中一個原因呢，就係話我哋去揾一啲新藥呢，未必有好好嘅位點去揾或者去標誌啦。咁其中一樣嘢就係話喺啲臨床前嘅研究係我哋冇一個好好嘅模型係可以仿效到人嘅身體啦。咁而譬如好似我哋一啲動物嘅模型啦，講緊係一啲嘅 zebrafish 啦，講緊係動物啦，或者係一啲嘅動物模型啦，咁老鼠啦，都未必係代表到人嘅身體啦。咁變咗我哋一開始實驗喺嗰度做嘅話呢，咁可能好多嘅藥呢係喺動物嗰度有用，咁但係就去到人嗰陣時呢，係會係失敗啦，因為始終個生理係唔同嘅。咁而其中令到我哋嘅一個挑戰呢，就係話我哋去用一隻藥嗰陣時，其實我哋好多時都唔知嗰隻藥會點樣影響個人嘅。咁而數學上我哋係講緊兩樣嘢啦。第一，我哋唔知道用咗藥之後對於嗰個人或者對於嗰群嘅病人有啲乜嘢嘅典型嘅效果啦。咁第二，亦都我哋唔知道喺呢啲病人嗰度每一個人嘅效果嘅唔同有幾大嘅變化啦，或者佢哋有幾大嘅唔同啦。因為我哋其實對於人嘅身體嘅結構啦，或者係個人嘅身體，其實到最尾可能都唔係話真係太了解啦。好多嘢我哋係未必知道啦。咁而雖然我哋喺咁而因為我哋呢啲嘅唔了解呢，咁係令到我哋喺揾一啲新藥啦，或者係用一隻藥嗰陣時呢，對於生理上嘅影響呢，我哋係都係好多嘅未知數嘅。咁我自己嘅研究啦，比較多係基因嘅研究啦。咁而啲基因嘅研究，希望係可以令到我哋對於我哋嘅身體、對於個生物系統係有更深嘅認識嘅。咁而講緊喺基因方面呢，當然我哋有好多嘅唔同嘅基因嘅排序啦。咁而每個人都係有一套唔同嘅排序啦，我哋有好多相似啦，咁但係亦都喺基因上有好多嘅唔同啦。咁而我哋每一個人都係有一個唔同嘅組成嘅。咁而呢啲嘅唔同呢，都係會令到我哋嘅生理上係有唔同啦。咁而我嘅理念呢係，So the idea is, in order to understand what the consequences of the drug changing that bit of our biology is, find individuals who happen to carry genetic variants that affect the same aspect of our biology, and just look at what happens to those. 咁而我嘅諗法呢就係話，用一次藥嗰陣時，其實我哋想知道佢點樣影響到我哋嘅生理啦。咁而對於一個基因學嘅學者呢，我係希望可以揾到一啲基因嘅變異呢，係會唔會對於佢生理呢係有相同嘅變化？咁即係話，其實我哋係喺揾翻一啲基因嘅數據啦，係揾到呢啲基因嘅數據點樣影響到我哋嘅生理啦，咁從而係對於我哋嘅生物嘅表徵啦，或者我哋嘅臨床嘅數據，或者係嘅成個身體嘅系統啦，係有一個相同嘅變化嘅。咁而其中一樣好有用嘅嘢就係話，好多基因嘅變異呢，對於我哋嘅生理呢，都係好重要嘅。 咁但係過程當然係好多挑戰啦。咁其中一樣嘢就係話個數據好大啦，咁變咗我哋喺分析上係要有好強嘅能力啦。咁而呢個數據呢，或者呢個大數據呢，除咗基因之外呢，係包括咗幾樣嘢嘅，譬如話一啲基因嘅表達啦，即係話喺 RNA 嘅層面上嘅一啲嘅數據啦。蛋白嘅數據啦。咁而我自己除咗話喺大學嘅工作之外呢，我都係做咗一間嘅創科公司啦。咁係希望將呢啲嘅數據呢，帶到去商業嘅層面嗰度，譬如 Genomics PLC 係呢一間公司啦，咁係我自己有份去創立㗎啦，咁係希望係將呢啲嘅數據啦，係包括咗一啲嘅超過1000個唔同嘅基因嘅研究嘅數據啦，咁係入面有500萬個唔同嘅人嘅同埋有超過5000個唔同嘅臨床嘅情況啦，咁啊希望將啲數據整合咗咧，咁而係令到我哋係可以係真係應用到呢啲唔同方面嘅組學嘅數據。咁而其中一樣嘢就係基因上啦，咁入面嘅數據咧有1400萬個唔同嘅基因嘅位點，咁嗰啲嘅位點可以係有一啲嘅變異啦，或者係有啲嘅唔同啦。咁又係知道呢啲嘅變異咧係好多咧係會影響到我哋嘅生理嘅表徵嘅。咁而我哋呢啲嘅表徵咧，可以有一啲係疾病啦，有一啲係佢哋嘅生理嘅數據啦。咁而有好多嘅可能係一啲實驗室嘅數據，係代表住嗰個人身體嘅唔同嘅情況。For any one of those 14 million, you can think of us as having a big matrix, so 14 million columns, one for each of these positions. SNPs, they're called, that vary. 咁你可以想象到呢1400萬個基因點啦，係一個好大嘅數據啦，因為每一個點嗰度都係有唔同嘅數據。咁而我哋係希望將呢啲數據咧，同埋一啲嘅呢5000個唔同嘅生理嘅表徵咧，係串合埋一齊嘅。咁而希望係用呢啲數據咧，係可以幫我哋預計到當一隻藥應用喺一個人嗰度，可能會出現到嘅變化。咁而我而家就做一個例子啦。咁啊，呢間就係我創科公司做出嚟嘅網站啦。咁而大家可以睇到一啲嘅基因位點嘅變化啦，係同一啲生理資料或者表徵嘅變化嘅關係。咁大家可以睇一睇呢度啦。
74 million, etc., along the chromosome. And it's a variable position. Variable position. SNPs have names. This one's called, not very exciting names. This one's called RS12916. And it's near a gene, and that gene is called HMG co-reductase. So many of the clinicians amongst you will know that that's the gene which is the target of statin drugs. So those drugs have been around for 20 or 25 years now. They've had a substantial impact in lowering LDL cholesterol levels. And as a consequence of that, they've had a massive impact globally in lowering the burden of cardiovascular disease and lowering mortality from cardiovascular disease. So this is a case where actually we know about the drug and we know what it does. Now, what I'm going to show you is that if we use the idea I've described, if we find a genetic variant that does something that's a bit like the drug, and look in people who are carrying that, that's like they've been on a version or a weak version of the drug all their life, what can we see? So this particular genetic variant, humble name, RS12916, amongst other things, has the property that it changes expression of this gene, kind of dials it up or dials it down a bit in the liver. So statin drugs inhibit this gene, HMG co-reductase, that's like dialing it down a lot. the variant that's plotted here, the alternative allele, if you change from a T to a C, then that actually dials it up. So the way it's plotted here is the opposite direction from the drug. And what does this picture mean? So each row here is one of those things we've got measurements for in people. In fact, we've got many different studies of cholesterol, and most of the rows you can see are about cholesterol. These circles are telling you what the effect of the change from the T to the C, so what's the effect of dialing the gene up a bit. And the bars show our 95% confidence interval. And so the fact that all of these circles and bars are away from the null hypothesis, the line that says there's no effect, are saying that if you look at individuals who happen to carry a genetic variant which dials the gene up a bit, does the opposite from the drug, then that increases their cholesterol levels. Or the converse of that is if you look at the other individuals, the individuals who carry the variant which dials the gene down a bit, that's like individuals who've been on a weak version of the drug all their life, they have less LDL cholesterol. So it's one of those instances, as I said, we know now that the drug works, but had we had this data 30 or so years ago when the drug was being considered, we'd have been able to say, actually, this idea suggests the same thing. Most of these measurements are cholesterol levels. One of them is heart disease, coronary disease. We now know that LDL cholesterol is causal for heart disease.
咁大家可以見到呢個例子啦，就係講緊一個單核酸多態性對於膽固醇嘅一啲影響啦。咁而頭先嘅例子嗰度咧，見到喺呢個基因變化嘅位點咧，咁係對於呢個 HMG-CoA 嘅轉化酶嘅表達係有一個影響啦。咁有啲可能係有啲可能會有啲人可能會做得多啲，有啲人少啲啦。咁而係除咗話對於呢個酶嘅表達係有唔同之外咧，咁而從而咧，我哋知道呢個酶其實喺我哋膽固醇嘅製造啦，或者係嗰度係有一定嘅影響啦。咁而因為咁樣咧，佢亦都係影響咗我哋心臟病、心血管疾病嘅一啲嘅機會。咁可以見到我哋喺唔同嘅方面嘅數據咧，都可以係睇到呢一個基因嘅變化對於嗰個轉化酶啦，對於膽固醇嘅level 啦，同埋對於心臟病、心血管病呢個機會有啲乜嘢嘅影響？咁係希望將呢啲基因信息同埋頭先喺嘅唔同程度或者層次嘅一啲數據係可以將佢哋整合埋一齊，而做到一個關聯性嘅。It's causal for it. One more example again. 咁另外一個例子啦，咁俾大家睇一睇啦。
Genetic variant, it's called RS2228145. It's on chromosome 1 at position 154 million and something or other on the chromosome. And the two letters that people have on their chromosomes at this position are either an A or a C. So again, what does this variant do? It's near a gene called IL-6R, so that's the receptor for something called interleukin-6. It's a key part of the immune system. The receptor sits on the membrane on the outside of cells, and it's used to signal information from the inside of the cell to the outside that the immune system uses. And there is a drug available. A number of companies have the drug, so Roche have a drug called tocilizumab, which blocks that receptor. It stops the receptor from working. And that drug improves outcomes for people with rheumatoid arthritis. So this genetic variant, again, is another example where it does something that's pretty similar to the drug. It happens to affect the way proteins are made and cleaved. And if you have the C variant here, then that decreases the amount of receptor you have on the cell membrane. So the drug blocks the receptor, so it means there's less that works. This drug means that this genetic variant means that there's less to start with. And so it's a bit like the drug. What do you see? Well, one of the things you see here is that those individuals who have the genetic variant that's a bit like the drug get less arthritis. So we know the drug works for arthritis. You also see they get less inflammatory bowel disease. They get less Crohn's disease, less ulcerative colitis. So that suggests this drug might also be useful for that condition.
咁喺呢個例子咧，可以見到呢個基因嘅多態性嘅位點啦，咁係會影響到IL6呢個基因嘅表達啦。咁而譬如話佢嘅一個位點嘅一個單核酸係會令到呢個 IL6嘅表達係細咗，就好似我哋有嘅一啲IL6嘅一啲嘅抗體啦。咁而我哋知道 IL6抗體喺類風濕性關節炎係可以幫到呢個病嘅。咁而我哋驚嘅數據同我哋講亦都係嘅，因為呢個位點如果令到 IL6係低咗嘅話咧，咁見到我哋係喺呢個類風濕性嗰度係有一個保護嘅作用。咁但係更加重要嘅一樣就係話我哋嘅驚嘅數據同我哋講話，除咗保護咗類風濕性關節炎之外咧，對一啲炎症腸炎都有幫助嘅。咁所以其實我哋可以估計到呢隻藥可能喺腸炎嗰度都會幫到呢啲腸炎嘅病人。咁呢個信息係令到我哋可以幫揾到喺同一隻藥入面嘅一啲新嘅用處啦。咁係希望係可以幫到藥廠啦，或者幫到我哋啦，係知道喺發展一啲新嘅藥，或者係有對一啲已有嘅藥係一啲新啲嘅用處。
This is the glucokinase receptor. This kind of colored set of wheels means the genetics says that if you mess with this gene, all sorts of stuff happens. So it would say if you're a drug company, probably don't go near that. This next gene, PLA2G7, so there's 0 here, means the genetics suggests that if you change that, you won't see any difference. That's another interesting example. There's something else I've talked about, LDL cholesterol. Something else you can measure in someone's blood is something called LPPLA2. And it had been observed epidemiologically that individuals with higher levels of LPPLA2 had higher levels of cardiovascular disease. Again, you can't tell from that whether it's causal. If it were causal, it would be worth intervening to lower levels of LPPLA2. Now, we didn't know then whether it was causal. One drug company, GSK, thought it was worth having a go. So they developed a drug which targeted this gene. This is the gene that produces LPPLA2. To reduce levels of LPPLA2. What would we have said if they'd come to us and said, what do you think would happen if we intervene and reduce levels of LPPLA2 via targeting this gene? We would have said, probably nothing's going to happen. Well, we weren't around, and they didn't ask us. They went ahead and did the experiment. They spent $880 million to do a clinical trial to see what happened when you lowered levels of LPPLA2. What did they find happened when you lowered levels of LPPLA2? Nothing. So that's an example where the genetic evidence would have stopped a very expensive clinical trial. So now I want to swap again and give you a sense of the other thing I promised. So those examples are about using genetics to understand how to develop better drugs, which targets to choose, and what the consequences might be of targeting those. Now I want to switch gears and say a little bit about individual variation and how we can make more sense of that with the range of data that's becoming available, not just genetics. So if the technology works. This is an iPad, and I'm about to show you an app that's on the iPad, if it all works. Great. Oh. Yep. Okay. So this is something, again, that we've developed, taking forward our academic ideas in the Spinout company, not really as a product as yet, but just to give an illustration of what might be possible. So the background here is, Simon mentioned a resource called UK Biobank, which is a study of 500,000 people in the UK on whom there's a lot of information, including information, it's an anonymized study, but information about their medical records and about genetics and many other things. So we took that as an example of what healthcare systems will look like in the future when all of that sort of information is available, and we used it for a number of purposes. So these are the data that's being used in the app is data on real people. We've made up names for them. We don't know their actual names. They're anonymized. So, for example, what this screen shows is for one individual, the one that whose name we've made up as Natalie, there's information which is accurate about her age, sex, height, and so on. And then what's shown here are predictions based just on genetics of what diseases she's at particularly increased risk of in the top portion and at decreased risk of compared to the average in the bottom portion. And she happens to be at higher risk of some disease based on her genetics than others. And actually if you can see these grayed out histograms, that's kind of the population distribution of risk. And you can see obviously in these examples, this individual's at high relative risk and in some cases high absolute risk as well. Now, one of the key points I want to get across, most people, when they think about precision medicine and think about genetics and genomics and knowing someone's DNA sequence, they think in terms of prediction. If you know the DNA sequence, you'll be able to say that person is going to develop type 2 diabetes, this person is going to develop arthritis, and so on. We're not very good currently at predicting outcomes based on genetics. We'll get better at that as we get more data. But a key message I want to get across is it would be crazy to do prediction just based on genetics because there's all sorts of other stuff we measure routinely in medicine, and we should use that for prediction as well. And I'll come back to that in just a moment. But you can scroll through individuals in the app and look at their differing degrees of risk for different conditions and so on.
咁呢個例子我哋可以見到啦，我哋係用咗一啲基因嘅數據啦，咁係可以我哋用咗呢個英國嘅基因資料庫啦，咁係可以將佢嘅基因資料係同佢哋有某種病嘅機會係連合埋一齊啦。咁而我哋揀咗一個人，知道佢哋嘅基因排序之後咧，我哋可以係預計佢唔同嘅病或者嘅表徵嘅一啲機會率啦。
All the stuff that any one of us has, we've inherited from our parents and grandparents and so on, and you can trace those back in the past, and it's relatively straightforward to predict for large chunks of people's chromosomes whether those chromosomes most likely come from an ancestor with European ancestry or with Asian ancestry or with African ancestry. And then what you can do, having done that prediction, you can then kind of colour in the chromosomes. That's what's shown here. And this individual has a genome which is fairly mixed. So the kind of light bluey colour is European ancestry, the greeny colour is East Asian, and the brown colour is African ancestry, and they've got ancestry from all of those parts of the world. This individual, again mixed, but in this case largely European and East Asian. Someone who's mainly European. These are all UK individuals, so we'd expect a lot of European ancestry, mainly European. This individual is also mainly European. This individual is actually really interesting. We don't know anything about them because of the anonymization, but we do know from their medical record they suffer from something called sickle cell disease. So that's a condition that's endemic in areas where there's malaria, in particular Africa and some parts of Asia. It's not something that's prevalent in the UK. So this is an individual who self-reports as Caucasian, so white European. We don't know, but I can just imagine the clinical odyssey they would have had as a young kid who had a range of symptoms. Probably a typical primary care physician in the UK wouldn't recognise the symptoms of sickle disease because it's not something they have to treat very often. If they did, they would probably rule it out because the person has clearly European ancestry. Eventually it was diagnosed. If you look, you can see just on the end of chromosome 11 and in one other place, there's a tiny bit of the chromosome where their ancestry is African rather than European. Now that happens to be exactly the part of the genome where the beta-globin locus is and where the sickle mutation is.
咁呢個例子咧，其實就係話我哋可以用一啲嘅基因排序啦，知道大家嘅族譜啦，或者大家嘅家族喺邊度嚟啦。咁而其中一個例子就係話，雖然佢係一個佢自己話係一個英國人啦，咁佢係一個外國嘅白人啦，咁但係就佢有一個叫做鐮刀型嘅紅血球貧血症啦。咁而正常其實喺白人嗰度呢個病其實好少嘅。咁而我哋做咗呢個基因排序，發現原來佢有一部分嘅基因咧，佢血統咧係喺非洲嗰度嚟嘅。咁而正因為呢一個 佢呢一部分嘅基因係喺一個非洲嘅血統啦，咁而解釋到點解佢係白人，但係就亦都得到咗呢一個喺非洲或者亞洲先至常見嘅一個遺傳病。
What we should be doing is using genetics and all the other stuff we measure in the clinical records. And that's what we did. UK Biobank allows us to do that. There's information collected on individuals of the sort that would be collected in healthcare systems and information about hospital admissions. So we kind of played a fairly standard statistical game. We've actually got data on these individuals up to 2017. We pretended we only had data up to 2012 and used that to predict what would happen over the next 5 years. You take some of the individuals, there are 500,000 in UK Biobank, so you take some of them and use that to fit a statistical model, use a machine learning type algorithm, and then you take other individuals and you use that to check that you're not overfitting. So we did sensible statistical things. This is actually, in some sense, the simplest of those prediction models that you could do. And again, for each of the individuals, you can say, using all of that information, what are the conditions at which they're of increased risk? And Hector, again, is at increased risk of MI, of a heart attack. And you can even look, in his case, to see what are the risk factors he has that's increasing his risk and which are the ones that aren't so relevant to him. We've even used the idea of colouring those risk factors because from a clinical point of view, some risk factors are important for prediction. If you're male and 70, then you're at higher risk of an MI than if you're a 50-year-old female. But those aren't things a doctor can do anything about. There are other risk factors where intervening would be important. And again, that comes back to causality. So you can look not just at individuals, but you can look across a whole cohort. So these are those predictions across all of the individuals. And if you want to, so that might be interesting from a public health point of view. You could say who are the individuals at increased risk and have a look at them and pick out their individual risk factors.
咁呢個係用咗一個英國嘅好大嘅組織銀行啦，咁唔係話淨係基因資料嘅，咁係擺咗入去，其實有好多關於流行病學啦，關於佢哋嘅一啲基本嘅資料啦，或者臨床嘅資料啦，咁係用咗啲資料係幫我哋預計到好多病，咁而我哋係可以展望啦，未來可以係用過呢個基因啦，或者係臨床嘅資料咧，咁係幫到我哋去實踐我哋個人化嘅醫學。咁最後咁我都喺度總結啦，咁係歡迎大家去問好多嘅問題。
Such a system is being under development, but as you pointed out in your last statement, that to make such a system work, you first of all have to have quite a sizable population, and I think probably you need to have people from different ethnicity or different background in order to have the generalizability of your data. And secondly, it depends very much on how much information that you can gather from the patient's clinical file and how long you follow up so that you know the outcome in your prediction of the 4,000 or 5,000 phenotypes that you have. So, so I would say this is a project forever in the development and evolution. How and when you think the system is mature enough to be really applied to drug development or prevention of diseases that I mean, you mentioned a couple of genes that may have either one phenotype or many phenotypes being affected when the gene is perturbed. So when would that actually you become confident that this outcome is actually predicting the correct thing?
No, it's a really good question. Let me take it in two parts. One of them is the bit about drug development and choosing targets, and the other one is about the kind of individual prediction. There's already evidence from two studies, one from GSK and one from AstraZeneca, which shows that even rudimentary genetic information in support of a particular target doubles the success of that target in clinical trials. So that changes the 10% to 20%, and you may think 20% is not very good, but actually just that change would radically alter the economics of drug development. Now, I think the setting we're at the moment, we're already able to be much more sophisticated than that. So I think those sorts of genetic ideas to find or validate drug targets are ripe for use now, and in fact our company has an announced collaboration with Biogen, a large biotech pharma, to find new targets in multiple sclerosis for them. We're at the early days of that, and I think these approaches, and I think in particular the ones we've developed, can make a big difference. There'll be a point in the future, maybe it's 5 years off, maybe 10 years, when no one would think of developing drugs without this kind of information at their fingertips, because it gives us a way of learning about what happens in human biology in humans. So I think that part's pretty easy on the prediction side. Yeah.
What about people from different ethnicity? We talk about potential collaboration between the 3 cities or the 3 universities. Do you think if we can gather the biobank material from Hong Kong or Macau combined to what you have, primarily English patients, would enrich your database and make it more generalizable?
Absolutely. Those sorts of studies would. China Kadoorie Biobank is another very good example where there's a lot of potential there. I think one point to make is that when, in the first part of my talk, where we're talking about developing drug targets and working out what the typical response is, you might learn something in a Caucasian population that could be helpful for developing drugs that will work everywhere in the world, or you might learn something in an Asian population that would help develop drugs that would work in a European context, because we're learning about the consequences of genetic changes on human biology. I think, exactly as you said, Joseph, when we come to thinking about tailoring this to individual, whether it's clinical support tools of this kind or prediction, that's a much harder problem. It's one where variation across groups, because of ethnicity, because of different, through genetics or different cultural factors, diet and lifestyle and so on, will be much more significant and much more important. So I think that's much further away. I think we'll get to some stage in the future where these sorts of techniques, all the data that's collected and statistical or machine learning approaches will start to be useful clinically. When that day is, I'm not sure. 5 years, 10 years, 15 years?
But if a chimpanzee's gene is only 1% different from the human gene, how different is the British chimpanzee from a Chinese chimpanzee, or a British man from a Chinese man?
Well, it's interesting. So the genome's the same, and almost all the genes are the same, and you'll know, and maybe many in the audience will know, actually the set of genes we have is very similar to the set of genes that a mouse has, and it's not that dissimilar from the set of genes that a fly has, and it's got quite a lot in common with the genes in a yeast cell. So that's like it's an analogy that's been used before, that's the building blocks. What makes us different from yeast, flies, and chimpanzees is the ways in which the building blocks are used and how they're turned on. So that's all of the machinery about regulation. And that's clearly the stuff which differs significantly between different species and between us and chimpanzee. Again, I think the key idea is this idea that if we use genetics, we can, it's been called nature's clinical trial, we can look at genetic variants which have particular effects on our human biology. And then if you have the right kind of data, you can see that if you change this A to a T, it increases expression of this particular gene. That changes the level of that protein. You can also see changes on other proteins, so they're probably things that are happening downstream in pathways. Some of those we'll already know about, some of those we won't know about. You can then see consequences on other molecular phenotypes, cellular phenotypes, all the way through to biomarkers and disease outcomes. So I think those things will be helpful, and much of that is learning about fundamental human biology. And my claim would be we can potentially learn about that in an Asian population and learn things that will be useful for all humans, or learn about it in a European population. There'll be some genetic variants which are more common in Europe, so that approach will work more easily there. There are others that will be more common in this part of the world, and it will work more successfully here.
Okay.
Peter, I think one of the complexities is that oftentimes with gene mutations, we don't know if it's going to be a loss of function mutation or a gain of function or no change or a de novo function. So for these SNPs that you're using, is that known? Is it known what direction it goes in? And does it even matter?
It absolutely matters. And I mean, it matters anyway in terms of our understanding of biology. And if you're developing a drug, you need to know whether you want to inhibit or agonize or antagonize the target. So direction absolutely matters. Most common genetic variants which have an effect on biology have an effect not through changing the protein which is produced, but they have an effect through affecting regulation of the gene. And as we develop the right kinds of information, so information on gene expression and so on, we're getting better, actually both experimentally, we're getting much better at studying gene regulation directly, but also computationally, we're getting much better at making those predictions. So it's not true in the case of every variant, but there are already enough variants where I can be confident that if you change this A to a T at this position, then in these tissues, it will increase expression of that gene, and that'll have these consequences on proteins and so on.
Okay, I think this will be our last question. Yes.
Hi, Professor Donnelly. Thank you very much for such an inspiring talk. I want to ask if you discover an SNP on a non-coding region of the genome, a non-promoter region, and what would be your recommendation to in terms of regulating what they can do to combat this particular change if it is a non-coding SNP?
So I think I understood the question. Let me try and answer, and then you can tell me if I've answered the wrong question. So most of these genetic variants, most common genetic variants, have only small effects on people. So the example I showed you on cholesterol levels, that RS12916, it's close to my heart, so I remember its number, RS12916, that has a small effect on the expression of the gene, and it has a small but measurable and real effect on cholesterol levels. But in order to see that effect convincingly, you need to study 50 or 100,000 people. The effect on cholesterol is small of that genetic variant. It's useful, I think, because it can tell us what happens if you perturb human biology in the same direction as the drug. The drug, statins in this case, has a much bigger effect on the gene, that has a much bigger effect on cholesterol levels and in turn on heart disease. So if someone happened to have the C rather than the T, or whatever the example was, variant at that position, you wouldn't be worried about them as a clinician. That will have a very small, so that having the C rather than the A or whatever at that position will have a tiny effect on their individual cholesterol level and not in a way that would increase heart disease risk. So you probably don't want to try and intervene to correct that in that person. I mean, there are hundreds of these common variants across the genome now known to affect LDL cholesterol. If you had the wrong version of the variant at many of those positions, then collectively they might start to have an effect on cholesterol levels, and you may want to intervene. You'd probably be, depending on their age and so on, you'd probably be monitoring cholesterol anyway, so you would have done it empirically on the basis of the cholesterol level rather than on the basis of the genetics. Did I understand the question? Yeah.
Thank you.
Okay.
Okay, so with that, I think we need to close this session. So let's thank Professor Peter Donnelly in this usual manner. So following this session will be the tea break, I suppose. Yes.
Yes, I believe that we can abandon our ID card, but we never have to use the fingerprint no more because this is already Stone Age. But please don't forget your iPad, or at least your cell phone should be anytime with the battery. Otherwise, you will be wiped out by the modern technology.